My name is Elizabeth Stevenson, and I'm a PhD candidate in Jay Dunlap and Jennifer Loris's lab in the Geisel School of Medicine at Dartmouth. And today I'm very excited to tell you about our recent review that was published in BioEssays. This work was a collaborative effort amongst members of our laboratory who study circadian compensation. Our review synthesizes the current models for circadian compensation amongst the most commonly used model organisms in circadian biology. Circadian rhythms are cell-autonomous biological oscillations that occur on a time scale of around 24 hours, and they enable organisms to anticipate the daily environmental cycles that are inherent to life on a rotating planet. Circadian rhythms are present across all kingdom and most eukaryotic phyla, and they work to regulate many physiological processes, including metabolism, behavior, and sleep. The molecular clock in animals and fungi is comprised of a transcription-translation feedback loop in which the positive arm of the clock, a heterodimer of transcription factors, activates transcription of the negative arm of the clock, which then feeds back to inhibit the activity of those positive elements via phosphorylation. In contrast, the bacterial clock does not rely on transcription or translation and can be reconstituted in vitro in a tube. This clock is driven by the oscillation of phosphorylation of two key phosphosites on the protein chi-C, which is regulated by the opposing forces of the proteins chi-A and chi-B. The plant clock is a distinct and complex transcription and translation feedback loop with many interlocking feedback loops that regulate one another, primarily through repressive action, in which cohorts of genes expressed at different times of day repress each other and one another. Circadian rhythms are defined in part by their ability to maintain a consistent period length despite changes in the ambient environment, including temperature and nutrients. This capacity to buffer against environmental changes is termed compensation. Compensation is a critical feature of circadian rhythms that maintains the moment-to-moment speed of the clocks across day-to-day environmental changes. However, the molecular mechanism of this fundamental property is still not clear. Our review covers the current models for both temperature compensation and nutritional compensation across mammals, insects, fungi, bacteria, and plants. In mammals, the temperature compensation model relies on a phosphoswitch on PER2, the key negative arm protein, in which two regions on PER2 have an opposing effects on PER2 stability when they are phosphorylated, and the rate at which these regions are phosphorylated depends on the temperature. However, the true consequence of these phosphorylations on PER2 are still being debated. The temperature compensation model in Drosophila derives from the mammalian model and proposes a similar phosphoswitch, but there is also evidence to suggest that the nuclear export and import of the negative arm protein TIM also impacts temperature compensation. In the filamentous fungus Neurospora crassa, temperature compensation is likely maintained through activity of kinases, including casein kinase 1, casein kinase 2, checkpoint kinase 2, and likely glycogen synthase kinase 3. Besides GSK3, the action of these kinases is on the negative arm of the clock. In cyanobacteria, temperature compensation is maintained entirely within the actions of the chi-A, B, and C proteins, with most of the evidence suggesting that temperature compensation is driven by the inherent temperature compensation of the ATPase activity of chi-C. However, recent evidence is beginning to develop the temperature compensation model further to include temperature influence folding of and regulation by chi-B. In plants, temperature compensation is primarily regulated by the action of the morning loop proteins late elongated hypocaudal, LHY, and circadian clock-associated 1, CCA1. Many of the clock mutants in plants that alter compensation ultimately lead back to changes in one or both of CCA1 and LHY. All roads lead back to these two proteins. In addition to temperature compensation, the clock is also compensated against changes in nutrients as well, although this nutritional compensation has received less attention than temperature compensation. In comparisons, many temperature compensation mutants have been found to have normal nutritional compensation, and many mutations that cause nutritional compensation changes have wild-type temperature compensation. This suggests that while mutations of a few proteins may impact both aspects of compensation, that there are very likely distinct regulatory mechanisms underlying these two aspects of compensation. In mammals, period length is maintained across changes in the amount of serum that is supplemented to cells in culture. In both Neurospora and in mammals, it has been found that perturbations of certain genes, such as the histone methyltransferase SETD2 or the polyadenylation factor CPSF6, to cause the period length of cultured cells to be impacted by altered levels of glucose or changes in other nutrient conditions. No nutritional compensation mutants have been identified as of yet in Drosophila, but there is evidence to suggest that the period length of eclosion, which is circadianly regulated, is not impacted by changes in protein concentration. Just as is the case with temperature compensation, the bacterial clock in vitro is also nutritionally compensated, suggesting that the nutritional compensation mechanism can be maintained entirely by the proteins chi-A, B, and C. While the plant clock is not compensated against changes in carbohydrate levels, it has been found to be compensated against changes in salt levels, and there also may be interplay between the clocks of the plant microbiome and the plant clock itself. Compensation is present even across very divergent circadian systems, from cyanobacteria and plants whose clock architectures are very distinct from that of mammals, flies, and fungi. This indicates that compensation is a non-negotiable aspect of the circadian clock. Circadian rhythmicity does not exist without it. The presence of compensation across these very diverse circadian models also suggests that there is not just one way to compensate a clock against environmental changes. Our review enables readers to compare the current observations of circadian compensation across all these disparate organisms. Thank you!